All right. Hi, folks. My name is Andy. I'm the research lead at the MCIU team, the team that's building the Human Reference Atlas. I'm giving the talk today, but these slides were put together with heavy input from Bruce, our technical director, Yash, our research software engineer, and our PI, Kadi Berner. Good to see everybody here. I'm going to talk to you today about the Human Reference Atlas, or HRA. Concretely, we're going to talk about the web user interfaces we make available, the HRA API that allows you to access HRA data in a scalable and programmatic manner, and also Jupyter widgets that we have implemented to use our applications in your Jupyter notebooks, including workspaces. Just briefly about the HRA, we put out a Markov paper last year in Nature Methods. I put the link into the chat, so feel free to read up on the HRA there. But briefly said, the HRA is a multi-scale and multimodal and three-dimensional atlas of anatomical structures and cells in the healthy adult human body. And it provides standard terminologies and data structures for describing specimens, so donors, tissue blocks, but also biological structures and spatial positions linked to existing ontologies. So we try to connect the HRA to existing units of knowledge out there in the world via these ontologies. It's a very central part of the Human Reference Atlas. The paper got quite a bit of attention on social media and the press. I just put a little collection here. And so today I want to specifically focus on the HRA applications that allow you to access HRA data via a variety of channels, depending on your level of familiarity with coding. So we're going to look at four concrete ones. We're going to look at the exploration user interface, which allows you to spatially and semantically explore tissue blocks and data sets inside of the 3D human body for male and female, 77 organs. We're going to look at the Cell Distance Explorer, or CDE, which allows you to visualize and analyze cell-to-cell distances in 2D and 3D tissues for codecs, SciSif, and other modalities. We're going to look at the HRA API, which again allows you to access HRA data in a scalable and programmatic manner. And finally, we're going to look at Jupyter notebooks and how they can be run inside of workspaces. And I'm going to anticipate a little bit there the part about workspaces at the end. So consider that a little spoiler. Brief. So basically, the talk is divided into three sections. We're going to have a high-level section. So if you have very limited coding experience and you just want to use HRA applications in the web browser, we're going to look at these interfaces running in the web browser. Then we have a mid-level section where we're going to run some API calls in our web browser. So most of this is going to be coded live. I have some backup slides in case it doesn't work, but I want to do all of this live. And then we have a low-level section where we're going to talk about how you can use our HRA API client, the Python version in this case, and also the widgets in the Jupyter notebooks and also in HubMap workspaces. All right, so a brief introduction to these user interfaces. This is one that we're not going to cover today, but it's important for understanding some of the other ones we're going to show. This is the registration user interface, which allows you to 3D register a tissue block inside of one of 77 3D organs. In this case, this is the female left kidney. You can see the user dragging a yellow tissue block towards the upper pole of this kidney. And so far, about 2,500 of these registrations have been made. And then these are all viewable and explorable inside of the exploration user interface, or EUI. So you can see here we're zooming into the visible human male. You can see the lung here with all of these many tissue blocks that have been registered. And these tissue blocks include many, many tissue blocks by HubMap with direct links to the HubMap data portal, which I will demonstrate here in a second. But it also contains data from ZenNet, GTEx, and other efforts. And then another interface that we're going to look at today heavily is the Cell Distance Explorer, or CDE, which again allows you to analyze and visualize cell-to-cell distances in 2D and 3D for a variety of different imaging modalities. Here's what this looks like in motion, but again, we're going to look at this live in just a second. All right, so this is demo time. I am first going to go over to the exploration user interface. So you can see that here in the middle, we have our 3D pane, which shows a variety of organs. We can see the heart, the spleen, the left and right kidney, as well as the skin. And then we have a feature here called spatial search, which I'm going to demonstrate briefly. So if I click on this, I can select my organ of interest. I'm going to pick the left female kidney. And then the organ is loaded here on the left side together with all of these existing tissue blocks that have been registered by various teams, including HubMap teams, inside of this organ. And then in the middle, you can see this golden sphere here. This is our so-called probing sphere. Now, the probing sphere can be moved around inside of the organ with my keys. And then if I make the probing sphere size bigger, say like this, it's going to take a second, and then we run a collision detection algorithm on the back end. And then it colors all of the tissue blocks that this probing sphere intersects with. So all of the tissue blocks that are within a set radius of the centroid of the probing sphere, they get highlighted in blue, and they also get named here. And then you can see here that these are the data sets and tissue blocks that are currently intersecting with it. So you can see there's some data sets here that we have already detected. So I'm going to click on apply. And this then applies this filter to the exploration user interface here. And you can see here that this is now reset. So we have captured 48 tissue blocks in this female left kidney. And then if I click on, for example, this one, this is a HubMap data, a HubMap tissue block. I can click on source data, and that takes me straight to the HubMap portal. So one of the goals of this talk is to show you how you can get from the HRA to the HubMap portal, what these kind of pathways are. So this then takes us to this particular donor with this particular metadata registered by the Vanderbilt team, I believe. All right, so that is the spatial search in the EUI. The next thing I want to demonstrate is the cell distance explorer. In the cell distance explorer, when you get to the landing page, and I put the link into the chat, you can either explore an existing 2D or 3D data set, or you can create your own. I'm going to create my own. And what you have to provide here, all you need to provide is a CSV file that has a bunch of columns, X, Y, and Z. Of course, the Z column is optional, and a cell type. So I'm going to we have a sample file here that I'm going to use. So I'm going to upload the sample CSV file. Here we go. And then I can add additional metadata for this visualization if I want. And then I'm going to click visualize. And then we get greeted by this screen here, where we have a three-dimensional pane in the middle that allows me to, in 3D, with an orbit cam, navigate around all of these cells. So every dot corresponds to a cell, concretely to a nucleus. And then we have these links that show that link that cell to the closest, in this case, endothelial cell in the rest of the tissue. This is also called anchor cell, but you can set any other cell type to the anchor cell type. And then on the right side, you can see some auxiliary visualizations. This is a violin plot with an inlaid box and Bueckle plot that shows you the distances, or also the average, median, etc. distances from, for example, T helper cells to the anchor cell, which is the endothelial cell. And you can see how these differ across different cell types. And then the same thing is visualized in the bottom with this histogram here. Now, these visualizations are made with Vega Lite, and you can actually download those visualizations as PNGs or SVGs, and then you can use them in your papers right then and there. Same thing goes for the visualization in the center. All right, this is the CDE. And again, all of this right now, we're still in the high level phase where we talk about how you can use all of this just by running it inside of your web app. Next, we're going to go over to the portal. So, and I'm going to get to questions at the end, by the way. I see there are already some questions in the chat. Now we are on the HubMap data portal, and we are on a so-called organ page. This is the organ page for kidney, where we get a ton of goodies from the portal. And from the HRA, we provide this organ information widget. So in this case, we have the left male kidney selected. So I can, again, use my orbit cam here and navigate around this 3D reference organ. We can see all of these different registrations that have been made into this reference organ by different HubMap teams. And this is only HubMap data, which is why it says only one consortium. And then you can change this to female and right on the spot. Many of these organs have male and female. Some have male, female, left and right. Some have only male or only female. There are even some that have only female and left and right. So there's various different combinations here, but the interface handles that for you. And so you can explore all of these registrations in 3D right there. Okay, so this finishes the high level portion of this talk where we just do stuff in our web browser. Now we're going to go to the mid-level section where we're going to talk about our API. So our API or application programming interface, we have a documentation page for it that allows you to run API calls and get responses in the browser. And I use this myself every day for all of the development work that I do. So I'm going to go here as an example to the aggregate results route. I'm going to open this. And then we have documentation of the query string parameters that I can use to customize my query. Now, this particular route is supposed to just give you the same kind of results that you would see in the exploration user interface. So what you can see up here with the summary statistics. And remember, this is already filtered based on the spatial search filter I was applying earlier. So go down here and we're going to click this try button here. And then I get my response. This actually went pretty fast. I get my response status. And so we have 18 consortia, so data from 18 consortia in the HRA from 49 tissue data providers. That includes, for example, the TMCs in HubMap, 724 donors, 38 organs, 2,135 extraction sites that link to 2,638 tissue blocks. Some of these tissue blocks share extraction sites. And then the assorted number of sections and data sets as well. And of course, this includes all of the HubMap data sets that have been registered in 3D with our registration user interface. And as you can see here, if I briefly scroll down, you can see that we have a big variety of different routes that you can use. Some of them include the usage of ontologies. So if you have specific ontologies IDs that you're interested in, you can simply punch those in and then it's going to give you HRA data based on the specified ontology ID. All right. So this ends the part about the API. We're going to be using this API inside of our notebook and then a workspace here in just a second. Okay, next up, I would like to talk about this table here. So the HRA is usable via the API in your own applications, but we also make these web applications available inside of Jupyter notebooks, concretely in the form of widgets. And to demonstrate and document the usage of these widgets, we made a series of HRA notebooks, and two of them, the ones that are labeled template, we actually make available as templates inside of the HubMap workspaces. And this is the part where I spoil a little bit of the fun that's going to be happening in later sections of this workshop where we're going to be introduced to workspaces themselves. So I made these slides available in the presentations folder. And you can see here I added Google Colab links to each of these demonstration notebooks. I want to just go ahead and briefly run a few of those just to show you the usage of our HRA widgets inside of these notebooks. So I want to start with the Cell Distance Explorer one. So I'm going to open this here. We are now inside of Google Colab. So this particular notebook demonstrates the usage of the CDE, which I was demonstrating in the web browser earlier, but now inside of a Jupyter notebook. So right when you do your analysis. So for example, if you do your data set analysis and you have spatially explicit data with an X and Y coordinate, or maybe even a Z coordinate, you can embed the CDE right there. And you can use that for doing your analysis. You can also do it for making figures for your paper. You can export these figures at the end as PNGs or SVGs. So I'm going to just run this. And then you can see we need some Python native packages. And then here are the import statements for the HRA Jupyter widgets and IPy widgets, which are dependency for this. What you can see here is that we have these widgets for a variety of, let's see here, scroll back up again. We have these for a variety of different applications. I was already showing you the EUI. The EUI organ information is what I was showing earlier on the HubMap portal with the organ page. We also have the node dist.vis, which is basically a cell distance visualization light. It's just the 3D part. Sometimes that's all that you want. And so if I scroll down, you can see that all we did here is we downloaded the sample file that I was using earlier in the CDE to make this very visualization. So we're just downloading that CSV, that file here, that Google Sheet as a CSV file. We read it in as a data frame. You can actually see how simple this format is. So it's just X, Y. Again, Z is not required. It's optional. But of course, you can also, if you have a two-dimensional data set, but you know how big, how thick your sections are, you can actually just write Python code right then and there to add a virtual Z dimension in there and just manipulate the data frame right then and there in Python. And then we have a cell type, which is just a string, but it could also be an ontology ID. It could be a variety of things. And then finally, we visualize everything with the CDE. So in this case, we have to do a small amount of transformations to get the data frame into the right format. Basically, it has to be transformed into a dictionary. And then our list of dictionaries, which looks like this. So basically, we transform every row into a dictionary. And then we have our constructor, the node Swiss constructor that takes a series of parameters. And then this is what we get. So this is interactive. It's running inside of my notebook. Again, this is just the 3D part. Now, of course, the CDE has a black background. Here, for presentation purposes, I chose a light theme, although I typically like to work with a dark theme. So this is just the 3D part. Sometimes that's all you need. Sometimes you don't need the auxiliary visualizations. And then if I keep going, we are using our CDE visualization constructor, and that actually gets you the full CDE widget running inside of your Jupyter notebook right there. We also get the auxiliary visualization on the right. So we get our box and Bueckle plots alongside our violin plots for the cell distances between all of the cell types and the anchor cell type, which again is the endothelial cell. And down here we get the corresponding histogram. You can, of course, also change what the anchor cell should be. And you actually do that right here. So node target value, in this case, I picked endothelial. But again, we could supply any other cell type that happens to appear in your data frame. All right, so this is this one. And then I want to also demonstrate our HRA UI widgets. And again, this one is a template in your workspaces. I'm going to show at the end of this talk how you actually get there. And I believe I have about another 13 minutes. I want to leave some time for questions. So this one here shows you, this is kind of a goodie bag that shows you a variety of different UI widgets and how they can be run inside of a Jupyter notebook. So I'm going to run this here. We have to install some dependencies again at the beginning. And then in this case, I'm going to be demonstrating 4 of them. Here I'm demonstrating the EUI organ information, which is the same component that you saw earlier on the, let me rerun this real quick, which is the same component that you saw earlier on the HubMap portal organ page. We're also going to look at the FTU Explorer Small that allows you to explore functional tissue units right there inside of your notebook. The medical illustration one, which is an FTU Explorer Lite, it just shows you the illustration and nothing else. And then also a model viewer where we integrated the Google GLB model viewer for web browsers. All right. So in this case, for example, we are looking at a functional tissue unit. So we specify an organ, which is the kidney, and then an illustration. This points us to a 2D illustration of a kidney renal corpuscle. We have 26 of those in total for a variety of 10 organs, if I remember right. And then this is what you get. So we use the medical illustration constructor here with the selected illustration, which is, as you can see here, this is a Perl that is then resolved to an actual SVG file, as you can see down here. And then we use this display function to actually show this. And then I can hover over these individual cells. We get a tooltip that shows us what the cell type is. That is illustrated here. And then another really nice thing here is I can actually capture user input. So if I click on one of these cells, for example, here the parietal epithelial cell, I actually get a console message here, cell clicked, parietal epithelial cell, and then I can also click on the same one again. I get a second message. So this is a way of kind of do some quote unquote debugging, console logging, what you see is what you get as you are working with this in your notebook. Then here is our organ information widget. So in this case, I just pointed the widget to the organ, which again is the kidney. And so then we have the female left kidney loaded here. This is of course interactive. I can zoom in, I can pan my camera, and I can rotate. So this is standard orbit camera control. And I again see now that we have 5 different consortia that have registered data into this from 8 different tissue data providers, etc. And then finally down here we have the FTU Explorer, which then loads the illustration, which is the renal corpuscle, alongside all of the other illustrations for the kidney and these other organs. And it then allows me again to inspect, hover over these individual cell types. It allows me to see source data that has been mapped onto these illustrations. And we're gonna have some updated data for this here coming very soon. But this allows you to use this interface right there in your Jupyter notebook. And then finally, this has been extremely useful for showing 3D assets inside of Jupyter. We made a wrapper around the Google Model Viewer. So you can point this to an existing GLB file. In this case, this is our visible human female left kidney. So the same thing that we saw earlier up here in our organ information widget. And now I just see the organ without any tissue blocks, but I can see all of the 3D structures and how this actually looks like with this kind of multi-point lighting here displayed in your Jupyter notebook. All right, we got about 8 minutes left. 8 minutes left. I do now want to go over to how you can actually run this inside of a workspace. So I am now here in my HubMap workspaces, and again, there's going to be other speakers later talking in detail about how to get there. But when you make a new workspace, you see that 2 of these notebooks that I was demonstrating are actually available as templates. Let's see here. Yeah, so you can see we have these HRA tags. We have the API tutorial, which introduces how you can make different API calls with our HRA client, HRA API client. And we also have this widget tutorial that I was just demonstrating. You can load this in there as a template. So this just makes it super easy to get started with using these widgets in your workspaces. Now, I do want to briefly show you another neat thing that you can do. I already made a workspace for this demo today, so I'm going to launch this real quick. You can actually just download a Jupyter notebook from Colab and then upload it into your workspace and then just run it there. So even if it's not available as a template yet, this is one way of just simply reusing the code that I was showing here with really very minimal setup. So this is taking a second here. So I'm gonna show this with some screenshots that I took. I was demonstrating the templates already. So one thing that you can do in Colab is you can simply download a Jupyter notebook like so. So you go File, Download, and then download the Jupyter notebook. And these are self-dependent, so you can just execute them basically anywhere. And then there's an upload button in your workspaces that allows you to simply upload this particular Jupyter notebook and then run it like you would any other notebook in your workspace. And then it creates all of the requisite folders on the go for you as needed. All right, with that, I want to capture one more thing, which is work from our brilliant colleagues at Caltech, who are part of our team. This is CellSam, which has been published in Nature Methods recently. It's a universal model for cell segmentation across different cellular imaging data. There is a code here that you can download as a Jupyter notebook. So if you go here, it opens this documentation page, and there's a gallery on here that allows you to download all of their demonstration code also as Jupyter notebooks, which you can then, of course, also import into your workspace and run there as needed. And then we have a similar setup for deep cell types, which was developed specifically for HubMap, which is a vision model that predicts cell types from different proteomic images. And there's likewise a documentation collected right here. All right. I do want to briefly advertise that we frequently hold HRA hackathons where we do basically what I was just telling you. So we're using our code, hacking our code to make it work for your data, build new applications, build new analyses with all of these widgets and notebooks that we have provided. The next one is going to be from March 9th to 11th here at IU. If you are interested in joining, please contact me so we can arrange for you to join. Or my colleague Bruce, who couldn't make it today. But we're very excited to welcome as many of you as possible here at IU in our beautiful Luddy Hall. And finally, I'm of course just part of a gigantic team led by Kadi Borner. And we have a variety of collaborators. HubMap is one of the consortia that we work with. And with that, I want to go over to some Q&A. So I did see some questions in the chat, and I see that Kadi already addressed them, so that is great. So Himanshu had a question about whether the distance is calculated between all the cells or if there are some radii of neighborhoods. Yeah, so that's a great question. The CDE has a parameter in its constructor that allows you to specify the max distance you would travel from cells to check. By default, that is set to 1,000, which is, I think, set to 1,000 microns. So there's a way to customize this. And depending on how low or how high you set this parameter, the computation is going to take longer or shorter. And there's a paper, I'm going to put that into the chat also, Himanshu, that details the Cell Distance Explorer that has more information about these kinds of questions. But yeah, great questions. Thank you.
Andy, did you run into any issues loading more than 1 million cells? Have you tried? I think it's Yash Moore who is working on the spatial omics side. You're working more on the Human Reference Atlas cell type population side.
Yeah, I have not tried that yet, but please, by all means, go ahead and try it. Try and see how far you can push it. And yeah. One thing that people have done is visualize chunks of data sets. That's also possible. You can pre-process and subsample before you use the CDE. You can also, it's going to run differently in a web browser than it will in your notebook. So it just really depends on the data set that you supply to it and the parameters that you supply to it also. It also, for example, depends on how many different cell types you have in there. If you have 10 cell types, it's going to give you a very different visualization than if you have 100 cell types in there. In fact, one thing I would recommend, if you have a lot of cell types, it makes sense just for visualization purposes to maybe aggregate them up to higher cell type level hierarchies, just to minimize the number of colors shown, just to make a more meaningful visualization. And this is something that you will find quickly once you start working with the tool.
And Andy, I would love to query the audience for who is actually doing spatial omics versus transcriptomics data. Maybe you could unshare your screen so that we see each other. And then people may be able to raise their hand if they work mostly with transcriptomics data, so by gene data, if you could do that. Or yeah, I think you can do it digital or you can show yourself. So transcriptomics data, so by gene data. Maybe that's not in the audience. How many of you are using spatial omics data, so spatially explicit data that you could actually read into the Cell Distance Explorer? They must be using different data, or they are all shy. I don't know which one. What other data do you use? Can you put it in the chat?
I guess we're also still at the beginning of the workshop. People might still need to use their shy. Okay.
Yeah, I mean, ultimately there is multi-omics data now also, and so like human cosmics and geomics and Meerfish, and of course that is also super hyper interesting for atlas construction. H&E images are interesting as well. I think sometimes you can, based on morphology, identify cell types, and then you could do cell distances between different types of cells that are identified in H&E based on morphology. But sometimes you also have codecs and matching H&E, at least in the HubMap data. And then you can run OMAP-based cell segmentation and annotation. And then again, you would have segmented out cells with cell types annotation. And then those are typically read into the Cell Distance Explorer. So spatial transcriptomics. Yeah, this is really important to understand also who is working on what data, is trying to do what kind of analysis or what kind of QA, QC. It's really interesting to capture that information. And thanks so much for joining us here today.
I see spatial proteomics codex data now by Daniel. Yeah, that's a prime use case for the CDE. And I mean, as you saw, like actually, if you go to the CDE website, let me share my screen real quick. One of the sample data sets is actually codex data. I'm going to share my screen here. So if you get to the landing page for the CDE, we have two existing use cases that are pre-made visualizations. So if I click on this one, um, oh, never mind, this is decisive, but it would work very similar for codex. So this actually gives you a pretty good idea of what your data could look like if you visualize it with the CDE.
And then you might have seen that HubMap has created over the 7.5 years many, many OMAPs. So we have 37 validated antibody panels, which typically speed up antibody panel design for immunohistochemistry-based assays. I put the link in the chat also. And it's good if you start with those and make minor modifications. The data that then results can be much easier used for atlas construction. Plus, you spend less time optimizing these panels. And again, these panels are all for healthy human tissue across all kinds of different tissue preservation methods and also organs. So we are happy to connect with teams that are interested to create other OMAPs. We're also working on an antibody panel builder that makes it possible to use existing HubMap, ZenNet, and other experimental data. And also, of course, the ASAT plus B tables that were created as part of the Human Reference Atlas effort and the existing OMAPs to help you understand what antibodies from which providers and which lot numbers might be best. There's a corresponding AVR database, which I can also put into the chat. And again, many, many people have spent a lot of time to create these antibody validation reports which ultimately help us get to an agreement of what are the best antibodies to use for which organs, for which preservation methods, etc. And hopefully speeds up the creation of good panels.
I have another question for the audience. What organ are you mostly working with? Really curious how the dice fall on this one. So what organ are you mostly working with?
Oh, many organs, and that's good. So we oftentimes want people to focus on, of course, one
Yeah.
organ, like the brain, complicated enough. But then it's also interesting to see teams now taking on multiple organs.
Colon is popular. That's unsurprising. All right. So the good news so far is that all of these organs are in the HRA. So we have 3D reference organs for each and every one of them. The adipose one is the most recent addition. We added that in December.
Yeah, great question.
Yeah. Then I have a follow-up question. So in the talk, we covered three tiers: high level using applications in the web browser, mid level using the API, and then low level using Jupyter notebooks. Based on your own work, which of these kind of levels or approaches to the HRA is the most relevant for you? High, mid, or low? Okay, low. Right. This is really good to know. This really helps us understand the users much better. Of course, it also stands to reason that many people here would be low level since this workshop is going to cover workspaces prominently. Excellent. Yeah. So you folks are the people that we try to support by making all of these widgets and making them easily accessible inside of notebooks. All right. Are there any other questions from the audience? In this case, Kadi, if it's okay with you, maybe we can hand the baton off to the next speaker.
Absolutely. Thank you, Andy. Great presentation. And again, reach out with questions if you have them.
Thanks, everyone. I'm going to be putting my email address and the link to our hackathon website into the chat. Thank you.